Hello everyone and welcome to our channel, MooTekey. It's wonderful to have you here as we dive into the fascinating world of chemistry. Today, we're exploring the essential concepts of electrochemical cells and reference electrodes. Get ready for an exciting and clear journey through the science of how chemical energy becomes electricity. Let's begin with the fundamental question. What is an electrochemical cell? An electrochemical cell converts chemical energy into electrical energy, or sometimes the reverse. It consists of two half cells linked by an electrical circuit and an electrolyte. You'll see electrons flow from the anode to the cathode. This flow is driven by redox reactions which generate what we call the cell potential, symbolized as E cell. In our diagram, we can see a voltmeter connected to a copper electrode in a copper sulfate solution. This half cell is connected via a salt bridge to another container with a zinc surface in a zinc sulfate solution. The salt bridge is a key part of completing the circuit. Now, let's get a closer look at the components overview. Here we have a detailed schematic of an electrochemical cell. We see the voltmeter at the top. On one side, we have the anode, which is where oxidation occurs, made of zinc, immersed in zinc sulfate solution. On the other side, we had the cathode, where reduction occurs, made of copper, immersed in copper sulfate solution. The two solutions are connected by a salt bridge, which often contains a salt like potassium chloride. The standard cell notation for this setup is written as zinc S, solid, versus zinc sulfate, aqueous, which is then connected by two lines via a salt bridge to copper sulfate, aqueous, versus copper, solid. To summarize, the anode is the site of oxidation, the cathode is the site of reduction, the salt bridge maintains charge balance, and the electrolytes act as ionic conductors in each half cell. As we continue, let's understand the anode more deeply. The anode is the site of oxidation, which means it's where a loss of electrons occurs. It's important to remember that the charge of the anode depends on the type of cell. In a galvanic cell, which produces electricity, the anode is negative. However, in an electrolytic cell, which uses electricity to drive a reaction, the anode is positive. The material for the anode is carefully chosen based on its ability to undergo oxidation and its electrical conductivity. Next, we explore the cathode. The cathode is the site of reduction, where a gain of electrons takes place. Just like with the anode, its charge depends on the cell type. In a galvanic cell, the cathode is positive. In an electrolytic cell, the cathode is negative. The cathode is often made from an inert material like platinum or carbon, or sometimes a reactive metal surface to facilitate the reduction reaction. Now, what about the connection between them? Let's discuss the salt bridge function. The salt bridge is crucial because it completes the circuit through ion migration. Inside the bridge, anions move toward the anode and cations move toward the cathode. This movement prevents the bulk mixing of the two different solutions while maintaining electrical neutrality. Salt bridges are commonly made with solutions like potassium chloride or potassium nitrate, held in place by a material like agar or a porous plug. Moving forward, we'll see the working principle in action. This diagram beautifully illustrates the electron flow through a wire from the zinc anode to the copper cathode. At the zinc anode, oxidation is occurring at the surface, turning zinc metal into zinc 2+ ions. Because it's losing mass, the electrode actually loses mass over time. The salt bridge shows nitrate ions and sodium ions moving to balance the charge. On the other side, at the copper cathode, copper 2+ ions from the solution are being reduced, gaining electrons to form copper metal on the electrode surface, which means the electrode gains mass. In essence, the two separate half reactions create a voltage difference. Electrons travel the external path through the wire, while ions travel the internal path through the solution. The cell potential is calculated as E cell equals E cathode minus E anode, and we use the Nernst equation for non-standard conditions. A load can draw current from the setup until the reagents are depleted. Coming up, we'll discuss reference electrodes and their role. Reference electrodes provide a stable, known potential against which we can measure the potential of another electrode called the working electrode. This enables accurate and reproducible measurements. Common types you'll encounter are the Standard Hydrogen Electrode, or SHE, the Saturated Calomel Electrode, or SCE, and the silver-silver chloride electrode. It's helpful to remember that an indicator electrode, like a glass pH electrode, responds to the analyte, while a reference electrode provides a stable baseline. Let us now look at the structure of the Standard Hydrogen Electrode, or SHE. This is the primary standard against which all other electrode potentials are measured. The diagram shows a setup with a copper wire connected to a platinized platinum foil, which is immersed in a solution of hydrogen ions at a 1 molar concentration. Pure and dry hydrogen gas is bubbled over the platinum surface at 1 atmosphere of pressure. A glass jacket surrounds the assembly. The half reaction here is 2 H+ ions plus 2 electrons being in equilibrium with hydrogen gas. By definition, its standard potential is set at 0 volts at 25 degrees Celsius. Now, what are the properties and use of the SHE? As the primary standard, it defines the 0 point on the electrochemical voltage scale. Its pros are that it's universal and reproducible, but this requires very strict control of conditions. Its cons include the difficulty in handling hydrogen gas, the risk of the platinum catalyst being poisoned, its bulky size, and the fact that it's limited to aqueous solutions. Because of these practical challenges, it's mainly used to calibrate other more user-friendly reference electrodes. Next, we explore the Calomel electrode, also known as SCE. The diagram shows a calomel electrode which contains a layer of mercury at the bottom, topped with a paste of mercury 1 chloride, which is in contact with a saturated potassium chloride solution. And you can even see potassium chloride crystals present. A porous frit at the end allows for electrical contact. Its notation is Hg versus Hg2Cl2 solid versus KCl saturated with chloride ions in aqueous solution. Its potential is approximately plus 0.241 volts relative to the SHE at 25 degrees Celsius. Its pros are that it's very stable and easy to use. Its cons are the toxicity of mercury and the potential for chloride ions to leak out, which is why you should avoid using it with chloride-sensitive systems. Now let's understand the glass electrode for pH measurement. This is the familiar bulb at the end of a pH meter. The diagram shows wires connecting to the pH meter. Inside the glass housing, there is an internal AgCl reference electrode immersed in a 0.1 molar hydrochloric acid solution. The key part is the very thin pH-sensitive glass membrane at the bottom. There's also a filling port, and the outer body contains a potassium chloride solution with another AgCl reference electrode connected to the sample via a permeable porous junction. This thin glass membrane becomes hydrated and develops a potential that is directly proportional to the pH of the outside solution. The change is about 59.16 millivolts per pH unit at 25 degrees Celsius. It's important to remember that the glass membrane needs to be hydrated to work properly and that measurements often require temperature compensation. To bring it all together, let's look at a comparison of electrodes. We have a table comparing 3 electrodes: the Standard Hydrogen Electrode, the Saturated Calomel Electrode, and the Glass Electrode. Looking at stability and practicality, the SHE has low practicality and is difficult to maintain. The SCE has high stability and practical for lab use. The glass electrode has good stability and is excellent for pH measurements. For safety, the SHE has hydrogen gas hazards, which are flammable. The SCE has mercury toxicity concerns. The glass electrodes is generally safer, though its glass membrane is fragile. Regarding the medium they work in, both the SHE and SCE are for aqueous solutions only. The glass electrode measures pH in various solutions. And finally, their primary use: the SHE is the calibration standard with E0 equals 0.000 volts. The SCE serves as a common reference electrode with E0 equals plus 0.241 volts, and the glass electrode is a pH measurement indicator. Other features can be referenced from the table for a deeper look. And finally, to wrap up our session, here's a quick summary. Electrochemical cells rely on redox reactions with separate pathways for electrons and ions. We have the anode for oxidation, the cathode for reduction, and the salt bridge to maintain balance. The Standard Hydrogen Electrode defines the 0-volt point, while the Saturated Calomel Electrode serves as a stable and practical reference for everyday lab work. And the Glass Electrode enables accurate and convenient pH measurement in countless applications. Thank you. See you in the next class.